Dear students, good morning to everybody. Hope you all are fine. Today's our topic is digestion of food. You know the process of digestion of food is accomplished by mechanical and chemical processes, and the digestion of food takes place right the time we put the food in the mouth, and this process ends in the small intestine. Now the digestion of food in mouth. The buccal cavity or the oral cavity performs two major functions, that is mastication of food and facilitation of swallowing. The teeth and the tongue, with the help of saliva, masticate and mix up the food thoroughly. Mucus in saliva helps in lubricating and adhering the masticated food particles into a bolus. Bolus, that is the mass of food. The saliva secreted into oral cavity contains electrolytes like sodium, potassium, chlorine, hydrogen carbonate or bicarbonate, and enzymes silvery amylase, ptyalin, and lysozyme. Silvery amylase is absent from the saliva of many mammals like cows and buffaloes, and predatory carnivores like lions and tigers. However, pigs have got amylase in their saliva. The chemical process of digestion is initiated in the oral cavity or buccal cavity. It is done by the hydrolytic action of the carbohydrate splitting enzyme, the silvery amylase. This enzyme hydrolyzes about 30% of starch into a disaccharide maltose at optimum pH of 6.8. The lysozyme present in the saliva acts as an antibacterial agent and prevents infection. Starch, which is a polysaccharide, is acted upon by silvery amylase at pH of 6.8 and is converted into maltose, which is a disaccharide. This mass of food bolus is then pushed down through pharynx into esophagus. This process is called swallowing or deglutition. This bolus further passes down from the esophagus through successive waves of muscular contractions. These muscular contractions are called peristalsis. There is a sphincter between esophagus and stomach called gastroesophageal sphincter. It controls the passage of food into the stomach. Gastric glands. The mucosa of stomach has gastric glands. Gastric glands have three major types of cells, namely mucous neck cells, peptic or chief cells, parietal or oxyntic cells. Number first cells, mucous neck cells, which secrete mucus. Number two, peptic or chief cells, which secrete the proenzyme pepsinogen. And number three is parietal or oxyntic cells, which secrete HCl and intrinsic factor and this factor that is factor essential for absorption of vitamin B12 in intestine. Now the digestion of food in the stomach. The stomach stores the food for 4 to 5 hours. The food mixes thoroughly with the acidic gastric juice of the stomach by the churning movements of its muscular wall and is called the chyme. The proenzyme pepsinogen on exposure to HCl gets converted into the active enzyme pepsin, the proteolytic enzyme of the stomach. Pepsin converts proteins into proteoses and peptones, that's peptides. The mucus and bicarbonates present in the gastric juice play an important role in lubrication and protection of the mucosal epithelium from excoriation by the highly concentrated HCl. Now, inactive pepsinogen in presence of HCl gets changed into active pepsin. Proteins are acted upon by pepsin and convert into proteases plus peptones. HCl provides the acidic pH at pH of 1.8, that is the optimal for pepsins. HCl of gastric juice converts iron Fe3 positive into Fe2 positive or Fe di positive in iron tripositive into iron dipositive, which makes the absorption of iron possible. Non-secretion of HCl, achlorhydria, or gastrectomy can lead to iron deficiency anemia. Renin is a proteolytic enzyme found in gastric juice of infants, which helps in the digestion of milk proteins. Small amounts of lipases are also secreted by gastric glands. The digestion of food in the small intestine, it will be inshallah in next offline class. Thanks.